be released by the basophils. So if someone went camping in the Rockies and drank water with Giardia in it, or some kid that was out playing in the sandbox and got a nematode, a worm infection, you'll see it increase proportionately in the eosinophils over other white blood cells. You don't have to remember all that information at the bottom of which tissues they're in and which tissues they're not in. I mean, it's interesting information, but I'm just saying you don't have to. I'm not going to ask you questions on that. Let me turn the. Pardon?
What did you say about asthma?
So it's going to go up in asthma because it functions to break down the histamine that's released in some allergic responses. So here you can see the really bright orange. Here it's got more of a pink tint. Same thing here. But you can see its relative size to the red blood cells. Again, these would be platelets. All right. So that pink to salmon color stands out pretty well. Now, the next cell is slightly smaller. All right, the basophil, around 10 microns in diameter. Now, compare this cell to our next cell. These are the two. This next cell is what everybody thinks is a basophil. So they see this dark blue and purple, and it can look very similar to this dark blue and purple. But if you look carefully, these have granules. And when you look along the edge, you can see an irregular surface as those granules are butting up against the plasma membrane. The lymphocytes, the blue, dark blue purple, is due to the nucleus, which has its own membrane and has a smooth surface to it. Can I erase this list? You guys have that already? So eosinophil, basophils are our next one. They're slightly smaller. They're just a bit larger than a red blood cell. Okay, a red blood cell is about 7 microns, 7.5, and these are about 10. Draw that a little bit smaller. Again, a bilobe nucleus. But now the granules are almost the same intensity and hue as the nuclear material. And so they easily obscure the nucleus. So large, coarse, dark blue staining granules. Remember, they can obscure the nucleus because this is the whole cell. This is not a section through a cell. So we're looking through the entire cell. But where the granules push up against the granules push up against the plasma membrane, that creates an irregularity that you can use to help clarify whether or not this is a lymphocyte. So approximately, what do I have, 8 to 10 microns, or just 10 to 12?
10 to 12.
These will secrete histamine. Do you guys remember the other cell, not a blood cell, that releases histamine? Mast cells. Okay? And the histamine increases the permeability of those postcapillary venules. And so more white blood cells can undergo diapedesis and squeeze through, and fluid can go through, and heat can go through. All right, so we see these in allergic responses. We'll be talking about antibodies again next week. But let's say that we have a particular foreign material in the cell, or in the body rather, and antibodies recognize that. The antibodies themselves don't do anything to it. They may clump it like they do the red blood cells, but a particular type of antibody class known as an antigen E is recognized by the basophil. It's not recognized by this specific component to it, but by the opposite end. And when the basophil binds to that opposite end, that triggers the release of histamine. So it's still considered to be a non-specific response because it doesn't require the specific recognition of the histamine, but the recognition of the type of antibody involved. Okay? So this is non-specific defense. And we'll talk about that on Tuesday, on how we group those forms of defense. All right, so these are our granulocytes. The other two are agranulocytes, not A and then granulocyte, but A meaning without. So the second most common cell is the lymphocyte. And this is what we're going to talk about next week, because the lymphocyte is our cell for specific immunity. This is where the B lymphocytes will make the antibodies, or the T lymphocytes will use chemical-induced cell lysis, the chemical being made by the T lymphocyte. And so it requires specific recognition before that attack occurs. It's not, the others are non-specific. So we'll look at T lymphocytes and B lymphocytes on Tuesday and Thursday.
I got extra pencils. Do you want to color something?
Pardon?
I was talking to Laura. She feels like coloring. I got extra pencils for her.
Taking notes. So 8 to 10 microns. I should let her come up and give a lecture. She's doing the micro part too. So 8 to 10 microns. They're slightly smaller than the basophil. So the red blood cell would be smallest, and then the lymphocyte, and then the basophil. So just slightly. Let me change the blue. Their nucleus is round and almost the diameter of the cell. So there's just a thin rim of clear cytoplasm that is present. And if you don't see that, you can see how that light, those changing shades of purple and dark blue would be confusing. No granulocytes though, so they can't be a basophil. You have to look carefully at that aspect. There are two types of lymphocytes, B and T, and in this form you can't tell the difference without some antibody tests that recognize protein differences, but they will behave differently. The B lymphocytes secreting antibodies you know as plasma cells. We found them in loose irregular connective tissue. And natural killer cells are a form of specialized T lymphocytes. So all I'm going to put here for function is specific defense. Meaning they have receptors on their surface that will recognize molecules as being foreign and then respond to that. One of the cells that helps them recognize this is our activated macrophage. So the macrophage is the largest. It's around 18 to 20 microns. I think that's the range I have on there. And its nucleus, these cells tend to be affected by the other white blood cells around them. So you can see the other red blood cells, you can see that they deform them. They push up against them, they can appear collapsed. So they're going to have various shapes, but they're very large. And they have a bilobe nucleus as well, similar to the eosinophil and basophil, but it almost looks like it's overlapped on itself, or it's more kidney shaped. So it's not a deep U shape. And again, a clear cytoplasm. So macrophages will convert or be modified in their mature form. Rephrase that. It's going to be a long day. Monocytes. To a macrophage. And these are definitely phagocytic. So macrophages from the monocyte and our neutrophils that's their most important function, is this phagocytosis. Part of the role, as you'll see next week, of a macrophage is to ingest the virus or the bacterium. Once it's ingested, it fuses with the lysosome, which breaks that down. And then what the macrophage does is it associates those broken down viral or bacterial particles with molecules on the cell surface. If those molecules match the receptors on the B cell or the T cell, helper cell, it activates them. And so we call that role an antigen presenting cell, or an APC. So macrophages will function as APCs. There's a couple of other cell types, the B cell, and a cell related to the macrophage, the dendritic cell, which comes from the monocytes as well, that function in that role. And we'll talk about that on Tuesday or Thursday. So I believe you have a quiz that ends tomorrow that asks for recognition of these types of cells, and may also include hemostasis, which we won't talk about till tomorrow. So I'll change that probably to Monday. Okay? I'll do it before I leave the classroom so I don't forget.